Good afternoon, everybody. My name is Karen Parr and I'm the CEO of BioMelbourne Network, and I very warmly welcome you to today's bioforum, Advancing Challenging Biologics, delivered by BioMelbourne Network in collaboration with ProBio. Let me begin by acknowledging the traditional owners of the lands on which we stand today, the Wurundjeri people of the Kulin Nation. I pay my respects to their elders past and present, and BioMelbourne Network is committed to supporting the future leaders. I also extend that respect to any Aboriginal and Torres Strait Islander people joining us today, both here and online. As we gather today to explore the frontiers of biologic therapies, we recognise the depth of knowledge, resilience and healing held within the world's oldest continuous culture. First Nation people have been the original knowledge keepers and healers on this land, and we honour their wisdom as we continue the collective journey of learning and discovery. And today's event is a continuation of that journey. This is a space for insight, connection and innovation. We are absolutely thrilled to see such strong engagement as well. Tickets have been sold out for a few weeks here. We have got almost 100 in-person registrations and 140 participants joining us online as well, so a special welcome to you. We have got people joining us from across Australia, but also internationally. So we've got Singapore, Hong Kong, Korea and Japan, and a very warm Wamanjika or welcome to you all. For those of you who are less familiar with BioMelbourne Network, we are Victoria's leading peak industry body for our thriving health technology sector. We proudly represent more than 200 companies, which is around 6,000 people, that are working across therapeutics, including biologics, medicines, therapies and vaccines, as well as medical technologies, so devices, diagnostics and digital health. Our members span from industry spin-outs and startups through to multinationals, from early stage companies through to manufacturing and in-market. Universities, researchers, investors, government bodies and the services sector that supports all of this important R&D taking place as well. This is a particularly exciting time for BioMelbourne Network. Not only are we celebrating our 25th anniversary next year, in just a few weeks, on Thursday the 19th of June, we are going to be launching our 25 to 28 strategic plan. It's a roadmap that builds on our strong foundations and it outlines our focus for the next 3 years as well. Our plan centres on connecting, amplifying and growing our sector through global engagement, strong networks, advocacy and a shared commitment to innovation, commercialisation and manufacturing. At the heart of our strategy is a commitment to cultivating a culture of connectivity and learning, and that is what events like today are all about. Offering high quality, thoughtful fora to support a sector-ready workforce and a globally competitive Victorian health technologies ecosystem. And it's why we're especially pleased to be hosting this bioforum in collaboration with ProBio, a company offering end-to-end CDMO services from drug discovery to commercialisation with proactive strategies, professional solutions and efficient processes in cell and gene therapies, antibodies and recombinant protein drugs, aiming to accelerate medicines development for customers. ProBio has established companies including in the US, the Netherlands, South Korea and China, and so I'm very pleased to welcome our guest speakers, including Dr. Ying Li, Director of Biologics Process Development at ProBio, who will be sharing her expertise and global perspective during today's discussion. This afternoon, we will be diving into the complexities and promise of advanced biologic therapies, including bispecific and trispecific antibodies, ADCs, and radiopharmaceutical drug conjugates. These cutting-edge innovations are reshaping the therapeutic landscape, yet they also bring challenges in design, manufacturability and translation to clinical use. You'll hear from leading voices across the biologics development spectrum, offering fresh insight into how strategy, science and collaboration can accelerate the path from lab to life. Thank you for being part of this important conversation. Before we begin though, I would like to invite the exceptional Dr. Rita Choueiri, Partner of Life Sciences at RSM Australia, to say a few words of welcome as our very wonderful and valued host today.
Hi everyone. For those of you who don't know me, I am Rita Choueri, a partner in the tax division here at RSM, leading the R&D tax and government incentives team. I'm incredibly proud to say that our R&D tax team is now one of the top 3 largest in Australia. I also head up the life sciences industry group, bringing my background as a chemist and my passion for drug discovery to the table. In case you're wondering, my PhD is not in tax, it certainly is in chemistry and science, so I do get asked that question. We are honoured to partner with BioMelbourne today in supporting this important event. I'd like to take a moment to warmly welcome all of you to our offices here at RSM. If you're unfamiliar with RSM, we are basically a full services international accounting firm with a global network. We rank as the world's 6th largest accountancy firm, and we're the 7th largest here in Australia. With offices in nearly every single country, we provide services to both local businesses and those with cross-border operations. We work extensively with companies in the life sciences sector, as well as in complementary sectors such as in manufacturing, technology, agriculture and so on, offering expertise across a wide range of services such as corporate and indirect tax, international tax and transfer pricing, external and internal audit, business advisory and general accounting, risk advisory, corporate finance, and in a range of other specialty consulting areas. And before I hand back over to Karen, I just want to express my deep appreciation for the work that this sector does. I was recently diagnosed with triple-positive breast cancer, and I'm now halfway through my chemotherapy and antibody treatment. Thanks to the incredible advances in the life sciences, I can stand here today and know that I will be okay. 20 years ago, that might not have been able to say that. The universe seems to have given me the full life sciences experience, and for that I can only be grateful. Grateful for the breakthroughs and for the research, for giving me the opportunity to be a better and stronger person, and for each one of you working to improve the lives of so many battling illness. And with that, I'd like to hand back over to Karen to introduce the next speakers.
Thank you, Rita. It definitely makes it more personal as well, so thank you. And thank you to RSM as well for being such wonderful and supportive hosts. It is now my pleasure to introduce our 2 speakers. Each will come up and present before our panellists then join them on our stage, where Dr. Lauren Pitt, Head of Preclinical at MirioTX, will lead the panel conversation. So our first speaker for today is Dr. Ying Li, the Director of Biologics Process Development at ProBio, leading the CMC process development and providing technical leadership in new technology platform development. She has led the process development for over 10 CMC projects with 7 IND approvals from the FDA and the NMPA, or the National Medical Products Administration in China. Prior to joining ProBio, Dr. Li was an R&D manager at the Merck Group from 2017 to 2022. She obtained her Bachelor of Science and PhD in Chemical Engineering from the National University of Singapore and the Pennsylvania State University, respectively. A frequent speaker at international conferences and author of 9 publications in top bioprocessing journals. Welcome. Our second speaker for today is Dr. Michael Wheatcroft, Chief Scientist at Telix Pharmaceuticals, who joined the company as one of the original employees. After completing a PhD focused on protein chemistry in the Department of Biochemistry, Cambridge University, Mike worked at the Cambridge Antibody Technology, or MedImmune UK, as it's currently known, which was a technology leader in the area of antibody engineering and protein sciences. After moving to Melbourne in 2010, he oversaw the preclinical development of several engineered antibody drug conjugates and the clinical translation of novel antibody fragment in prostate and ovarian cancer, including radioimmunoconjugates. Mike's worked in senior development roles at Medicines Development Limited, Hatchtech and Star Pharma, spanning GMP production, clinical study support and non-clinical studies for a range of pharmaceutical and medical device products. In his current role as Chief Scientist, Mike oversees both the non-clinical activities across the pipeline and leads the R&D team to explore new ways to enhance Telix's existing assets, as well as the development of new products and technologies. Please join me in welcoming Dr. Ying Li to the stage.
Thank you. Thanks, Karen, for the kind introduction. It's my great pleasure to be here and give today's talk on challenges and solutions for complex biologics CMC. So first of all, I'd like to give you a brief introduction about ProBio platform. Our company was actually initially founded in 2016 as business unit of GenScript Corporation and later as a spin-off in 2019. And over the years, we have accumulated extensive experiences in CMC development, obtaining over 55 R&D clearances globally, with most advanced projects has obtained emergency use authorization during COVID. And we have supported 2 marketed medical devices as antibody supplier and completed over 200 cell line development projects for R&D application and over 100 CMC and CMO projects. So over all the projects that have been done by us, about 50% are mAb molecules, with the rest of them complex biologics such as tri-specific antibodies, which count for around 30% of the projects, and 20% being fusion protein and recombinant proteins. Let's just take a closer look at the different modalities covered in our platform. So starting with bispecific antibodies, they can be broadly categorized by IgG-like, which contains the Fc fragment, and non-IgG-like contains Fab fragment. So under the IgG-like antibodies, we have done the symmetric bispecific antibodies such as IgG plus single-chain FV, and IgG plus single-domain antibody, and IgG plus peptide. And for asymmetric bispecific antibodies, there are a couple of examples with asymmetric formats with 4 different chains, 2 different chains, or 3 different chains. And we have done those type of molecules assembled in vivo and in vitro, such as the dual body format. And in addition, some examples include IgG plus cytokine and tri-specific antibodies. So under the non-IgG-like related antibodies, there are bispecific antibody fragments such as the Fab fragment, the VHH or VHH in tandem, and single-chain FV fragment. And other type of antibodies include the bispecific antibody conjugates, such as the SCFV conjugated in vitro through 3-step conjugation with the PEG linker and single-chain FV plus the Fab format. So on average, the titer delivered for bispecific antibodies is 6.7 gram per liter, with the average yield over 50%. And on the protein side, we have done a number of projects for R&D filings, including the IgG fusion protein, such as the Fab fusion protein and FC fusion protein. And also, we have completed IND applications for a number of recombinant proteins, such as the spike proteins for vaccines, the hormone, and some unstable enzyme and blood factors. And those molecules are currently under clinical trial, phase 1 to phase 3. And there are many unique manufacturing challenges associated with these complex biologics. For bispecific antibodies, the common challenges include the byproducts such as homodimer and aggregates and fragments, and low expression level and control. Recent years, we start to see the requirement for N-glycosylation optimization as well. And for recombinant proteins, common challenges include the low protein stability, how to design a capture step for proteins without tag, how to remove HCPs, improve expression level, as well as analytical method development. Significant efforts and time needs to be spent to tackle these challenges during the CMC development, and sometimes the solutions may not be effective. So over the years, we have established our toolbox platforms, building on our experiences for CMC studies to come up with effective solutions for these common CMC problems. And we have summarized this into our platform solutions covering different modules starting from cell line, cell culture, purification, formulation, and analytical development. And most recently, we are also incorporating AI into our platform database and hopefully creating smarter solution with AI supporting functions. So today, I'm going to walk you through a number of common challenges for CMC solution for these challenging molecules and provide our platform strategies and case studies in tackling these problems. So first, let's start with titer improvement. So how to get a high titer for our expression protein is one of the key tasks for the cell line development as well as the cell culture development. So the expression vector, starting with the cell line development, the expression vector is one of the key components for titer improvement. So different vector designs will directly determine the transcription and translation efficiency of the target gene, as well as the solubility, stability, and secretion level of our protein. So the key elements of a vector include a promoter, the signal peptide, as well as transcriptional enhancer. So I gave a couple of examples of our platform vectors. So our second generation UP vector implement a titer UP enhancer element, which help to improve the integration efficiency of the vector and hence significantly improve the titer of complex proteins. And this year, we also launched our third generation proprietary MAX vector. On top of the UP vector, we incorporate a stability enhancer element, which help to improve the cell line passage stability and help the cells to recover faster from the selection pressure during the stable pool stage. And here's just some data summary for change of the different expression vector and potential impact on the improvement of the titer expression. And also, speaking about the cell line selection, the high throughput monoclonal screening is one of the key technologies to enable higher expression titer. So the traditional methods such as limited dilution, all the FACS methods are suffering from low throughput and cannot be screening truly high expression single clones. And over the years, there have been emerging technologies such as VIPES, WOLF, single cell printer, and Beacon, and different instruments that help to improve the screening throughput and help to faster the cell line and select the high expression single clone. And this image shows our R&D team recently also developed our internal high-throughput clone selection platform, which utilize some fluorescent dyes to bind specifically to our target proteins and utilize microfluidic cell sorter to select the high-expression single clone. This image shows basically utilizing our new technology methods that help significantly improve the single clone for production. And moving on to the cell culture stage, there are a number of common strategies that could be adopted to improve the protein titer, such as the media optimization, including the feed, basal media, and additives, and optimization of process parameters during the bioreactor stage, and selecting different cell culture modes, such as perfusion or intensified fed batch. So those are pretty commonly used strategies to improve the titer through cell culture optimization. And here I just would like to highlight our in-house capabilities for in-house cell culture media development. So over last 3 years, our R&D team has come up with our in-house UProTro media, which designed specifically for our in-house host cell, and which has been tested in a number of projects and showed highest titer in 15 of 2022 projects and outperforming all the major media brands. And the average titer of mAb and bispecific molecule using our media, in-house media, can reach 7.6 and 6.3 gram per liter respectively. I think moreover, through this media development process, our team accumulated a lot of know-how on the potential impact of different media components on the titer and cell metabolism, as well as the product quality, such as how to control the charge variance, glycosylation patterns, etc., so that later in different type of CMC projects, we can come up with more customized solutions with adjusted media components to help improve the titer performance as well as particular CQA. And next problem we are going to talk today is on the homodimer control. So homodimer formation is one of the key challenge for the asymmetric bispecific antibodies. So here include an image just showing, for example, if we have asymmetric bispecific with two different heavy chains and 2 different light chains, if we allow this chains to assemble freely, the correct pairing of the molecule only count for about 10% of the overall expressed products. So different platforms utilize different designs to reduce the formation of homodimer. So here I list a couple of examples, such as the common strategies include like point or multiple point mutations in the CH3 domain on Fc to avoid mispairing of the heavy chains, such as knobbing to hole, the RTIG, which introduce electrostatic repulsions, and dual body, which is a controlled fiber exchange reaction, etc. And common strategies to reduce light chain mispairing include using common light chain or switch the CL and CH1 domain to get a cross-map structure to reduce the mispairing of light chains. So moving on to the cell line selection stage, our platform normally utilizes a dual vector system to improve the productivity and purity. For example, these are a couple of examples for asymmetric bispecific antibodies with 2 different parts, A and B. So we can basically construct all this sequence into one single vector. Or alternatively, we can utilize a dual vector system and put the A part into one vector and the other B part into another vector, and optimize the vector ratios in order to improve the overall productivity as well as reduce the percentage of homodimer. So here is one example. This is the format of the antibody with asymmetric structure. So initially, if we put all the sequence on the single vector, as we can see, the chain B will be significantly overexpressed. And as a result, the correct pairing molecule only count for around 22%, and the titer is at 0.5 gram per liter. So our team utilized this dual vector system and optimized the vector ratio and successfully improved the overall titer up to 5.4 gram per liter, and with the correct pairing of the molecule up to 72%. So if we cannot address the homodimer challenge in the earlier phase, we are moving into the purification phase. This is sometimes undesirable because we almost will lose a significant yield through the purification process. So sometimes this homodimer will be very challenging to be removed. How to tackle this problem, we will typically start with analyzing the difference in the physical and chemical properties of these two chains. For example, for this particular case, we performed the ICIEF analysis of the chain 1 and chain 2. And so we found out the delta pi between the two chains is at 0.94, which is quite significant. So this is a good indicator that we can potentially leverage some ion exchange chromatography to separate the two species. Here, I give one example here. Even with knob-in-to-hole structure, this bispecific antibody express 40% homodimer in the supernatant that has to be removed. And so upon analysis, we designed a mixed mode capital MMC step to remove this significant amount of homodimer, leveraging the difference in the charge. And here is another example. This is FC plus single chain FV fragments. In addition to the significant percentage of whole homodimer, there are also 20% aggregates that need to be removed. So we designed the CHT type 2. CHT mixed mode chromatography is always very effective in removing aggregates, and also designed the capital MMC process to reduce the whole homodimer. As a result, so the second step, chromatography, effectively remove the majority of the high molecular weights, and the third step, chromatography, remove almost all the homodimer, getting the final purity of the DS over 99%. All right, so the third question to be covered is around aggregates control. So broadly speaking, there are different types of aggregates, such as covalent or non-covalent aggregates. They can be reversible or irreversible, and soluble and insoluble. So there are a number of factors that contribute to the aggregates formation, starting with protein structure, the hydrophobicity of the protein sequence, and the secondary tertiary structure, and the way the protein is being folded, and post-translational modifications such as oxidation or deamination could all contribute to the aggregates formation. And also during the cell culture process, a couple of key parameters such as pH, DO, temperature, osmolar in the media, and redox condition in the cell culture could all impact the aggregates formation. And similarly for purification, different pH, conductivity, buffer, and freeze-thaw condition and shear force could contribute to aggregates. So in general, our aggregates control strategy, so people can come up with the purification process to remove the aggregates. Actually, the thinking process should start earlier with the molecule design as well as the cell culture optimization. So here is one example of optimizing the molecule structure to help improve the druggability of the molecule. This is a bispecific antibody with IgG plus single-chain FV, which is a very common format. So we know that the antibody with single-chain FV is known to prone to aggregates formation. So for this early phase molecule, we find out that during pre-CMC studies, we find out this molecule has high tendency to form aggregates. For example, under the wild type, upon concentration up to only 7 grams per liter, we can see a significant increase in the aggregates level. And so our team worked with the client to optimize the design of this particular ICFV domain, basically utilizing a longer linker between the C terminal of VH and N terminal of VL to help release the conformational stress on the VH domain, and also introduce a VH-VL interdomain disulfide bond to enhance the structure stability. So upon modification, we can see that the stability of the protein was significantly improved. So I think the pointer is not working.
Error message on it.
So in addition to molecular design, we can also potentially optimize the cell culture condition to help reduce the percentage of aggregates upfront. For example, this shows during the early phase study, during the protein expression, there's a significant aggregates formed, and particularly we can see that these aggregates only show up during the non-reduced condition, which give us a good indication that these aggregates are potentially formed by the disulfide bond mismatch and have been reduced during the reduced condition. So we designed the media screening process and conducted DOE study with cysteine, which is a reductant, to help break down the disulfide bond mismatch, as well as the temperature downshift during the cell culture, and significantly reduce the percentage of high molecular weights from over 90% down to 12%. And with respect to purification, the removal of aggregates is almost always one of the most common challenges in downstream purification, especially for bispecific and fusion proteins. So I just include one example with a Fab and SCFA molecule. This is quite challenging case. Over 45% of aggregates were formed upon production. So our team developed 3-step chromatography and utilizing the CEX step to effectively remove the majority of high molecular weights and getting the final purity over 99%. All right, so the last part is regarding the radionuclide drug conjugate. RDC is an emerging class of targeted radio pharmaceuticals for diagnostic and therapeutic applications. I think Mike is going to talk on RDC more in depth, but here I just want to maybe touch base on the CMC perspective of RDC development. So in general, the molecular structure of a typical RDC include antibody, chelator, and radionuclide. So there are different conjugation reactions that could be adopted for the RDC. For example, the commonly used strategy include the lysine-based random conjugation, and also cysteine-based random conjugation, or use engineered cysteine for site-specific conjugation, or enzyme-mediated site-specific conjugations. And there are a number of different chelators that could be selected targeting for different type of radioisotopes. And speaking for the lysine-based conjugation process, this is a very random conjugation process because we have like 70 to 80 lysine sites sitting in the IgG, and among them, around 10 to 20 of them are accessible for the conjugation process. And so here result in a very heterogeneous conjugation products. And therefore, we have to control the reaction conditions and optimize the different key parameters to acquire the target DAR value. So in general, our platform will adopt a one factor at a time study in the beginning, just to identify different key parameters that will impact the performance of the conjugation. For example, the molar ratio, pH, temperature, and the antibody concentration and reaction time for some cases. And here is one case study. This we have completed IND application for EMA and complete the GMP production for this client in Europe. So this is IgG molecule conjugated with DOTA, SCN, and we start with designing the manufacturing process for the monoclonal antibody and manufacture at 80-liter scale, and later develop the process for the DOTA SCN conjugation and further purification afterwards. And on top of the one-factor optimization, we also conducted a DOE study focusing on the number of key factors, such as the antibody concentration, the molar ratio of the drug versus the antibody, and reaction pH. And we come up with mathematical model that could predict the performance of different DAR value under different conditions in our design space. So as a result, actually, we conducted small-scale studies to validate our model, which shows very accurate prediction of our experimental DAR versus the predicted DAR using the DOE model. And we successfully scale up this RDC production with very robust scale up, as well as very robust DAR distribution across different batches, and successfully scale this up from 1 ml up to 10 liter GMP production. All right, so in summary, hopefully through today's talk, I have shown that we have extensive experience providing CMC services for complex biologics and establish our toolbox solutions to address these challenges. In the end, I'd like also to highlight that we have partnered with a number of biotech companies to assist our partners to complete a number of license out projects, including 6 license out projects, all covering different type of complex biologics such as bispecific and trispecific antibodies, fusion proteins, and ADCs. And so the example listed here, one of them is, I think the first one, this one was licensed out to GSK, and this was done back in 2021 with a very challenging trispecific antibody. And second case is also a bispecific antibody with PD-1 and VEGF targets, a pretty well-known deal, and licensed out to another MNC company in the US. And through this development process, we have accumulated a lot of very helpful experience supporting the due diligence as well as the manufacturing quality system built up for our company. With that, that's the end of my talk, and thank you very much.
So do I need the mic as well still? Good. Just check. Right. Thanks very much for the organisers. Thanks for inviting me to talk. I'm from Michael Wheatcroft from Telix Pharmaceuticals. Today I'm going to talk about sort of under the challenging biologics. We were a rare radiopharma company who developed both small molecules as well as biologics for radiopharmaceuticals. And certainly they have a few challenges, which I hope you'll agree at the end, but they're very much an emerging area of therapy in oncology and very much very highly effective. So, quick introduction to Telix, even though you've lost the world. So we're a commercial stage imaging, commercial stage company with Illuccix as our main imaging product for prostate cancer. And preparing more imaging products are on the way. We have a late-stage theranostic or therapeutic pipeline, including a very active clinical development. We have an advanced supply chain and manufacturing capability which we're expanding in recent years, and including manufacturing facilities now in US as well as EU. We're very much a global business, even though very proudly Melbourne-based, Melbourne headquartered. We've been, I think our 3rd and 4th employees were in US and Japan and then Europe. So we very much had that global viewpoint at the start. And today, through acquisition and building and just growth over the last 8 years, we're now up to over 1,000 people worldwide. So we're very much, and you can see here on the world map the number of acquisitions and new builds that we've had. So we've very much got this global presence from the start. So a quick introduction to theranostics. Often introduce them as very similar products in the way to antibody drug conjugates. In the way that they work, they both have they share a similar design brief, as I like to say. So they have you have a targeting agent on the a targeting agent of some format that binds the biology, the cancer target or the target you're looking to direct it to. You have some form of a linker, and then you have a payload, and so it's the same design brief as an ADC, really. But what is different, I guess, is obviously while you can still choose the targeting agent, but you also get a choice of the payload, and the payload in our case is a radiopharma or radioisotope payload. So that payload can be either a diagnostic radioisotope for use for imaging or a therapeutic one for therapy. And of course, your targeting agent very much dictates what the properties of that molecule, in terms of the route of excretion, the pharmacokinetics, as well as obviously that's the key element for binding. So that cancer specificity comes from that binding, that targeting agent. And obviously these agents, like ADCs, antibody-drug conjugates, are injected intravenously or systemically administered, and obviously it's basically a self-directing radiation therapy instead of often when you talk about targeted radiotherapy, or we talk about precision radiotherapy, often it means sitting in a machine and getting zapped by a machine. But this is very much a systemically driven drug where it can target the tumours just simply by the action of through biology. And as I mentioned before, you can either have an imaging modality, either PET or SPECT cameras, depending on the radioisotope you're using, or you can have a therapeutic radioisotope. And you can see very much the imaging modality here. It's very clear. It gives a lot of clinically valuable information in terms of the extent, the location of the cancer, and it's really kind of radicalizing and really changing oncology care, as we say. Even just the imaging that we have is really changing and saving lives in many cases. Moving on to theranostics, this whole area is being driven really in recent years by a class of molecules targeting prostate cancer, this target called PSMA, prostate-specific membrane antigen. And the small molecules have led the field in the early days in terms of being very simple molecules, and they seem to be relatively specific. And you can see the same in the chemistry, the same cancer targeting bit. It's basically just a dipeptide that is more like a substrate analog for the PSMA enzyme on the cancer cell. And you can see that there's a correlation here between the diagnostic imaging on the right and the therapeutic, which you can still image, but it's just not as clear. And you can see this one-to-one mapping of the cancer. You can see that you really are targeting specifically those lesions. So hence the term theranostics, because you're therapy and diagnostic in the same modality. So while small molecules are very well known, antibodies actually make great theranostics too. The field of antibody-drug conjugates has been driven through, no surprise, antibodies because of that highly specific nature, because it's essentially a native drug in drug format. So we're one of a few companies who's developing both small molecules and biologics. We recognise the power and specificity, and also from an industry perspective, the format, having a standardised format of drug delivery really helps as well. And you can see here some results from our phase 3 imaging study that was being completed, Zircon study. And on the left CT is what you could see there was the CT on the left-hand side was the standard of care. The black and white image was the standard of care for imaging. So it's basically lumpology, as some people like to say. It's very much you're looking at growths and different morphologies, but it doesn't tell you a lot about the pharmacology of that cancer. So on the right-hand side, you probably saw that great big blob. You don't have to be a nuclear medicine physician to recognise that that it's very clear, that it stands out like a bright light, and that's obviously the radiation targeting to the tumour. So I was going to say when I was looking at the talk, I have done a lot of protein chemistry in my life, have done some antibody discovering, drug development, and I was really looking at ways how I could fit this into the topic of my talk, into this complex biology. I haven't got enough data, as Ying kind of very professionally showed. But what I can talk about is a little bit about some of the complexities. Radiopharm is a sort of a different, it's got its own complexities, its own unique factors. While we share a lot with antibody drug conjugates, the radiation and having radiation as a payload really changes the nature of some things and makes you. So in some cases it's same, but maybe very much different also.
As an interlude, how does the FDA deal with a product like this?
It's a great question. I don't think they know. Yeah, that's a great question. So, like, as a radio label products have been around for a long time. Most of them have been diagnostic, and most of them have been small molecules. And so they've actually been handled under the Office of Diagnostic and Specialty Medicine. And so when you kind of bring along an antibody drug that's radio-labeled, they kind of look at it and think, does it go to SIBA as a biologic, or does it go to the CDRM? And the answer is they kind of both look at it, which means you get two different opinions on different topics. What's also interesting is with our biologics, we're also dosing quite low, very small amounts. So we're actually almost a microdose. In fact, our dose, there's many other drugs that use 1 or 2 milligrams of antibody, because you're not looking for a therapeutic effect from the binding. If you like, it's the payload and efficacy comes from the radioisotope. Again, I think FDA's constantly looking to modernize itself and keep up to date with trends, and in this case, that's the case there as well. Anyway, back to complex radiopharmaceuticals. So one of the key things and the key differentiators for radiopharmaceuticals is time and geography suddenly matter again. And a world where we could get everything, global supply chains, Zoom and video calls and everything, suddenly the idea of having to think about where your drug's manufactured and how you do it and the proximity to your patient actually kind of matters with radiopharmaceuticals, because you've got a time-sensitive product. It has a decay properties. It'll decay. Physics doesn't lie, and it doesn't matter. It doesn't care. When you're choosing your radioisotope, you're obviously looking at your emission profiles and your pharmacokinetics and the radiobiology of how it interacts with what you want to, with diagnostic or therapeutic. But you're also looking at you also end up determining your type of drug production, your drug delivery strategy, if you like, how you're going to get this to patients. Because radiopharmaceuticals are perishables, in effect. We often like to talk about the idea of a melting ice cube, that once you manufacture it, it's really, you've got a certain amount of time before it needs to be injected and used in a patient, otherwise it's no good. And so we talk about something called a shelf life, and it's not a shelf life in months or years, it's in hours and minutes sometimes. So, and that means the time in which the product is within specification from when it's manufactured and released. And that means both, obviously, you've got to account for the amount of decay that's going to get from where you're manufacturing it to where your patient is. That's the decay. But there's also, obviously, the effect of the radiation on your product. It can called something called radiolysis or radiolysis. It can affect drug product quality. So particularly because our biologics have a long biological half-life, so they swirl around the body for a long time, it actually matches really well with long half-life radioisotopes. So you think, great, I can manufacture in US and I can send it the other side of US or Europe, and we've got plenty of time to ship it. But also then you have to think about what the effect is on the actual product itself. So one of the challenges has been maintaining radio stability in terms of keeping your product in suitable conditions. So obviously that's primarily through formulation, conditions to extend that shelf life. Obviously looking at things like protein friendly, keeping the protein happy, like pH, osmolality, reducing agents, all those good things for your product. but also you're looking at stabilizer additives so even ethanol can actually reduce radiolysis, but also ascorbate or vitamin C ascorbic acid can actually act as a free radical scavenger and actually really help protect your product. So they're very common in radiopharmaceuticals. But even things like dose volume, the amount of volume of your dose and the vial shape and the vial type can actually, and as well as the temperature, can affect can impact your product, can help your product and save your product. But also, I guess the second thing you want to thinking about is also making sure you've got efficient delivery to the site that's used. So having a manufacturing network in the country or in close proximity to where your patient is, as well as an expert team of logistics and people to handle them as well. So there you go. And what I mean by that, just an example of different radioisotopes. This is a map of half-life on the x-axis here. And you can see there's two groupings. One is on the left-hand side is mostly diagnostic radioisotopes when the half-life are in minutes. So 6 hours for technetium, 110 minutes for F-18, and 68 minutes for gallium. So those are typically dispensed and distributed and manufactured through a radiopharmacy network. So that is typically within the same city or within the same hospital or within the same area anyway, the same area that you're making and dispensing. On the right-hand side, the longer half-life radioisotopes like actinium is one of the longer ones with 10 days. But for instance, lutetium or zirconium, down to even towards the 3 days, that's still a manageable time to be able to manufacture something and ship it and have it received for a radiopharmaceutical. And bizarrely enough, you might think this is just terrible commercial concept, but actually these radiopharmaceuticals have been zinging around for many, many years, and this is an established supply chain that does exist. This whole radiopharmacy network has been built up around doing this day in, day out from morning, noon and night. So on the right-hand side of these long half-lives, radioisotopes do match with this centralized manufacturing kind of capability. And just to give you a snapshot of that, on the left-hand side is the radiopharmacy network. This is actually a composite of some vendors as well as a radiopharmacy network we've just acquired last year. And you can see all the blue dots are all radiopharmacies, all distributed throughout the US, and they're obviously clustered around population centers. On the right-hand side is the map you saw before in a bit more detail, but you can see a number of those sites are manufacturing sites, so the ability to make your product and ship it to where it needs to be. Having that global supply chain, not of the drug product, not also of the drug product, but also the radioisotope as well matters. So challenge number 2, I guess, is this radiopharmaceuticals, these theranostics are not your average CMC campaign. Obviously, many people would be very happy to make a drug and put it into a blister pack and put it in a pharmacy, and have that used whenever it's needed. But in our case, where we've got multiple manufacturing steps, not just the biologic, which has its own challenges, as we all know, but also the bioconjugation. We have in the past had to use different vendors, so we have to release the biologic, we make that in bulk, and then bioconjugate in batches with a different vendor. So, and I think it's acknowledging now the move in the field towards the idea of having vendors that do both is really a game changer for us. Like ProBio is a manufacturer that does both biologic manufacture and bioconjugation, and it's a real game changer for us. So beyond the bioconjugation, you obviously have, I can't really see it, but the bulk manufacture is all done in advance. That's the product that gets fill finished. So actually there's there's the antibody production, bioconjugation, and then the fill finish, which is also another step, maybe a different vendor. And that's the product that sits in the shelf at the radiopharmacy or the manufacturing site waiting to be radiolabeled. And then it's radiolabeled on demand, I guess, for that patient or in production batches at the point of use. So what this means is you've essentially got 3 different products. So from a regulatory perspective, Jim, you'd like this. So the actual antibody and even the bioconjugate are still raw materials for us, as defined as a raw material in a drug product manufacturing process. And it's actually the first radiolabeling becomes a drug substance, and the final formulated dose that puts into a syringe or a vial becomes a drug product. So it kind of redefines a lot of things. And That also confuses some of our vendors sometimes when we call our antibody a raw material. Also, add in this the complexity of with bioconjugation, you also need your formulations to be metal-free, because you don't want random metals to be collated in your collator, your linker payload thing. And often this bioconjugation process needs optimization. A company like Telix has started off its life, very much the last few years we've been acquiring products, working with academic groups to advance their technologies. So we get a range of different chemistries and technologies that we want to carry on as best we can and show the same but different. But also that we need to kind of optimize them to make sure they're the best they can be, make sure they don't impact product quality, and also make sure that they're able to fulfill the yield. So one of these examples of bioconjugation is the legacy bioconjugation process we inherited from an academic group. And the chemistry actually worked beautifully for them. They were working in 50 mil Falcons, and they were able to change pH pretty quickly and do all these, run it through a PD10 column to do the purification, etc. But as soon as you start using a gram of protein, things become a lot more difficult, because you've got higher scales, bigger volumes actually really screw you up. And you can see this process, if you're any protein chemists in the audience, you can see this is basically protein abuse. Not just through the multiple adjustments and the number of steps, but you're pHing up to almost 10. You're adding a chelator that's solubilized in acetonitrile. And then you're changing the pH back down to 7, so right near the isoelectric point, using an acid. You're adding EDTA, then you're heating it to 35 degrees. And then you're changing the pH back again down to pH 4.4 and leaving it for 2 hours. So you couldn't really, other than microwaving and hitting it on the head with a hammer, you couldn't do much else worse than handling proteins. And even the formulation is typically they're very basic formulations with with radio labeling. So the formulation is just 0.9% saline with no buffer in. So obviously this creates challenge when you're looking at scale up, and particularly even the heating step. To heat up to 35 degrees, you have to heat it up slowly, and when you've got a litre of liquid, that becomes a challenge in itself. That caps your process yield. So we've taken over this process. We've been we've adapted this process in a number of ways. We adapted it primarily getting rid of that heating step by using a different transchelant. A different way of stripping out the unwanted metals from the formulation, from the linker. And that was a game changer in terms of not needing heating, not needing the low pH step. So automatically that was a way better process in itself. But then we've also applied an automation process to that. So automated pH adjustments, automated mixing, very standardized, reproducible, and that's exactly what regulators do want to see, is that reproducibility. So by going through that, we've reduced the product quality risk, reduced the batch, we sort of increased the batch-to-batch reliability, and we certainly get higher yields from it by doing so as well. And this is just an overview, I guess, of the manufacturing supply chain. As I mentioned before, we've got the raw materials on the left-hand side, the antibody bioconjugate, and then the drug product is the radio label part. Radioisotopes supply chain is a whole nother talk, because not all of it is accessible, and that's a whole subject in itself. But then in making sure that end-to-end logistics is there to deliver your product to the patient. They're also dangerous goods as well, because they're radio labeled. They come in massive heavy pots. So it has several challenges, but this is all something that is routinely done, and something we're tapping into. We're expanding that capability in network, and it's reinvigorating the whole sector because of it. Third challenge, I guess, thinking about radiobiology really matters. So what I mean by that is when you start thinking about your targeting vector and which radioisotope you use, having to think about a lot more about pharmacokinetics and delivery. You want, like in ADC, you want something that's got high targets, high cancer specificity. You want something with a high tumor uptake, ideally retention inside the tumor or inside the cancer cell, and you want to limit the exposure to healthy organs as you do. So, and with radiation, it's not like a drug falling off. You're not preventing a drug falling off like with an ADC. You're actually, it's going to be you're going to be exposing healthy tissue on the way through or in circulation or even just being stored in an organ, it will have an irradiation effect. And surprisingly, many organs are actually very radio tolerant, actually, and only it's about how radio tolerant that organ is that really matters, and which organ and where it is. So thinking about dose symmetry is very important for our drugs, which then couples to the time of targeting vector you have. So we've been looking at so antibodies, again, very highly specific. We like the specificity and the predictability of it, but it has a long circulating half-life. So naturally, you do get this exposure to the bone marrow, which is one of the radiosensitive organs. And that correlates, that's our dose-limiting toxicity, is actually the effect on bone marrow and hematology, so effect on blood cells. So we've spent a lot of energy looking at ways of optimizing this, and we've come up with a couple of different ways, one developed internally and one in-licensed, of next-generation targeting, biologics targeting. So the first one was a format we call RADMAB, which is full-length IgG antibodies, but they've been FC engineered to clear quickly. So the idea of that they deliver the dose, they get into the cancer cell, all the same properties that you like of an antibody, and yet it doesn't hang around as long. The second format we've looked is radiopharmaceutical mini-bodies, which are engineered antibodies that are smaller, faster clearing antibody fragments. And just one slide on the RADMAB platform. It looks and smells like an antibody, except a couple of point mutations that affect FcRn recycling, and it dramatically changes what we know as an antibody with a biology of a half-life of several weeks. It changes it into something that is cleared in a few days. And so it really does work. This has been taken into our phase 1 study in our 592 program, which is our prostate cancer therapy. And this is our alpha program. So we're looking at, with alpha radioisotopes, they're much more powerful, more strong, more highly potent. So we want to really control where it goes and make sure it doesn't impact the bone marrow. And again, that's undertaken its first phase 1 dosimetry study. It did exactly what we expected it to, and now we're excitedly pushing into a therapeutic program as well. So, and just mentioning about the mini-bodies, so we've been talking here on the right-hand side about IgGs as delivery vectors, and on the left-hand side, the other end, we've got small molecules and peptides, where a lot of companies are very much entered the theranostic space. And you can see they have different properties in terms of time it takes to get into a tumour, time it takes to actually clear from the blood, how well it's likely to be retained, as well as if it can be internalized into the cancer cell. So we have a range of opportunities in terms of delivery vectors, and the mini-body vector, which is sort of in the middle, is an engineered antibody format that has this balance of characteristics. So it's very much trying to get the best of both worlds, this approach. So what is a mini-body? I think Ian kindly showed before, but essentially it's an IgG that's had its guts ripped out in the middle, CH1 and CH2 domains taken out, if you like, and so you're fusing that CH3 with the variable regions to make a smaller version. Again, you've still got bivalent binding, you've still got, I guess, a recognizable format that can be manufactured, and importantly, the size of it is what makes the difference. In terms of the size of the kidney being close to the kidney filtration cutoff, means that it doesn't zip straight through the kidney and get lost like smaller formats like nanobodies or like single chains, and they're not big IgGs that get turned away, don't get excreted to the kidney. They have this slow excretion through the kidney. Which is actually quite handy because it means you can actually lose some of that unwanted radiation. You can actually control the excretion of that partly through two organs. It's being excreted through the liver as a normal antibody, but it's also being excreted through the kidney, and we can control how much of each it goes through. So you can actually change and modulate the pharmacokinetics, which is really nice. So you get this, again, we're talking about organ exposure. It's your kidney's getting a dose, but your liver is also getting a dose as well in terms of splitting the radiation between two. So that smaller size also drives faster tumour uptake, and we've got clinical study data that confirms that. It has fast clearance otherwise, and importantly, as a platform, you can take any IgG and with high confidence turn it into a mini-body. So it's a way of adding intellectual property. It's a way of life cycle management for some of our existing assets as well, or other people's assets. And here's the proof of the pudding. This is a clinical study that was run in Memorial Sloan Kettering Cancer Centre, and we really see that fast clearance, rapid tumour uptake, and we did see this split between the kidney and the liver, which is what we expected, which is what was expected. Hello again. This is my coming up to the final slide, so hopefully no more AV problems. But essentially, this shows the process of the evolution of the IgG conversion to mini-bodies. And so first of all, when you take away the CH3 domain, with the CH1 domain, you're no longer got that stabilizing force pinning it together as well. So it's important to have that packing between the two the heavy and light variable heavy and light chains. And so the idea of having that interface, having the right stable packing between those two, the heavy and light chains are really important. So that was the first generation, if you like, from more than over 5 to 10 years ago. And since then, the team at Imaginab who developed this technology, they also realized that you also need to stabilize the hinge, stabilize the structure of the antibody. And this was a patent-protected proprietary way of doing it that they found, a way of adding basically more stabilization. It really makes the product a lot more stabilized. They've also worked a lot with electrostatics in terms of trying to change its uptake or retention in kidneys as well. That's also, and again, a lot of proprietary know-how that they've developed in doing that. So in summary, targeted radiopharmaceuticals are definitely a new and emerging, I wouldn't say emerging, it's been around a while, but definitely expanding class of drugs. It delivers both therapeutic and diagnostic utilities. Small molecules and biologics can be used as targeting agents, but they have very different properties, if you like. And while there's a number of challenges and complexities, it's actually the recent clinical and commercial success of theranostics shows that they really are, it's worth it. Those challenges have been largely overcome, and these is very much now getting to that commercial stage of a technology. We talked about shelf life and the impact on radiation and drug quality, as well as complex manufacturing and the time sensitivity of delivering that drug to the patient. And as hopefully we've shown as well, is that antibodies engineered for radiation delivery, so designed for this new modality, really offer a new innovative option to deliver, to improve the therapeutic window for all of these drugs. And with that, I'll say thank you.
Michael, for those really insightful presentations. It's my great pleasure to be chairing the panel session today. And just by way of a brief introduction, I'm currently head of preclinical development of Myrio Therapeutics, which is a Melbourne-based biotech company, as well as Scio Therapeutics, which is a spin-out of Myrio Therapeutics. And for the past 3 years, I've been focused on evaluating the preclinical activity of Myrio's T-cell engaging bispecific antibodies that we've designed to target specific peptide HLA complexes on the surface of tumour cells. And prior to this experience, I was based in the US at a biotech in Philadelphia called Immunome. And there as Director of R&D, I led novel target discovery and antibody discovery and served on the leadership team. And I'm trained as an immunologist. I did my PhD here at University of Melbourne and postdoctoral studies at NYU School of Medicine in T-cell acute lymphoblastic leukemia. Just reflecting on my own experience, particularly at Immunome, where we were discovering novel antibodies and thinking about how to turn those into therapeutics, we would often state that we were open to different therapeutic approaches, but the one that we often turned away from was ADCs because we just thought the chemistry was too complex. And it strikes me now that Immunome is now solely focused on ADCs, which really speaks to the gaining popularity and accessibility of that technology. I'm really excited to welcome our panellists on stage. And before I do, I'll just give an introduction for the two additional panellists we have in addition to Ying and to Michael. Professor Andrew Scott leads the tumour targeting laboratory at Olivia Newton-John Cancer Research Institute, is director of the Department of Molecular Imaging and Therapy at Austin Health, and is professor at the University of Melbourne and La Trobe University in Melbourne, Australia. Professor Scott's clinical research interests are focused on developing innovative strategies for targeted therapy of cancer and molecular imaging and theranostics in oncology. And there have been 7 therapeutic antibodies developed in his lab that have been licensed to pharma and biotech, and 4 have progressed to clinical trials. Professor Scott has also been a lead commissioner of the highly impactful Lancet Oncology Commission on Medical Imaging and Nuclear Medicine, and the Lancet Oncology Commission on Radiotherapy and Theranostics. He's received many awards, including Member of the Order of Australia, election as Fellow of the Australian Academy of Health and Medical Sciences, the Lachman Award for Translational Research, the Saul Hertz Award from SNMMI, and Fellow SNMMI and Fellow of SNMMI. Professor Scott has published 485 peer-reviewed papers and 27 book chapters, is an inventor on 25 patents, and has received over $35 million in peer-reviewed grant funding in the last 5 years. We also have Dr. Jeremy Wu, who has over 20 years of experience in the pharmaceutical and biotechnology industry, holding research and leadership roles at Mimetopes, Star Pharma, and Avipep. He currently leads IDT Australia's ADC R&D program, focused on developing effective IDCs for cancer treatment. And Jeremy's extensive ADC expertise began during his time at Star Pharma in collaboration with the Baker Institute, where he worked on dendrimer antibody conjugates for MRI studies on thrombosis. At Avipep, he led the ADC program and worked with leading ADC developers including Immunogen, now AbbVie, Seagen, now Pfizer, and Spirogen, now AstraZeneca. His team advanced an antibody-based ADC to an advanced preclinical stage. Jeremy holds a PhD in chemistry from the University of Wollongong and completed his postdoctoral training at the Research School of Chemistry at ANU, and he's the author of over 20 scientific publications and co-inventor on 3 patents. So I'd like to welcome our panellists onto the stage to begin the panel discussion. Okay. I think we're sharing the microphone. Is there one on there? Oh, here you go. Okay, we might have to hand over. So I might start with you, Jeremy. From your perspective as the ADC lead at IDT Australia, are there particular challenges or opportunities you find especially compelling in this space?
Thanks. Thank you for the opportunity to share my experience and knowledge in ADC area. And actually, I found, I'm surrounded by all ADC experts. From the talk, the word I heard is challenges. So basically, for ADC, we talk about the challenges and opportunities. ADC from the concept of magic bullet by Paul Ehrlich, that is more than a century ago, and to the realization in the clinical use over a century. And for the challenges, we need want to just a brief history about ADC. So it started with the term, magic bullet, and then take about 75 years to have the mAb, because to that stage the mAb production was possible. And it take another few years for the first ADC in test. That was in 1983, I think. And then I think it take another 10 years, the first ADC using very cytotoxic reagents, calicheamycin, for the ADC. But that takes another 7+ years. By 2000, and the first ADC, Malotag, was in clinical use. But unfortunately, due to the adverse effect and not ideal therapeutic application, Malotag was actually withdrawn by Pfizer in 2010 fortunately, and in the next few years, we have Adcitrus and also TDM1 came into the market. And since then, ADC has been blossoming. So as of today, we have 17 ADCs in clinical use. And I'm very optimistic we will have many more ADCs being developed for clinical use. And but the challenge, we talk about this, it has taken more than a century to get 17 ADCs in clinical use. And also, half for the blood cancer, half for solid tumor, and the payloads very limited. And we think about only probably 4 big categories: Mitensin, and auristatin, calicheamicin, and camptothecin. Okay, only those 4 big categories. So ADC, like any other therapeutic agents, will develop drug resistance. So we need far more cytotoxic reagents to develop a variety of ADCs. So that's certainly a big challenge. But others, as also the format of antibody, for example, as Ying mentioned, bsADC. Actually, this is gaining pace, and it's considered as the next generation, really true ADC, compared with conventional ADC. And linking chemistry, releasing mechanism, we need to develop far more than current. Currently is very limited. Just use the opportunity to maybe share with you. The linking technology basically you have the acid-based, you have enzymatic release. So very few chemistries available. And hoping we'll have more. And this is our challenges, but the opportunities. We talk about this now. Last year, ADC market is $15 billion US, and it's forecasted in 10 years will be $115 billion. And in HER2, the blockbuster, we're talking about that, is $4 billion revenue last year. It's just only one drug. And bsADC, and the opportunities, I just learned the Chinese one made a deal with Bristol Myers Squibb, the single drug, $8.4 billion deal. It's a really mega deal. And upfront $1.3 payment. So now come to, we want to use the opportunity about, challenges in Australia. So I want to advocate, Australia's community for the ADC's involvement. So advance ADC for Australia. And our neighbors, China, is regarded as the leader, particularly in bsADC. And Japan is considered as a key player in ADC. Korea is growing very fast. So what about us? I think, we need, more collaborations, work together. So let's collaborate and to produce more results. So let's talk to each other more and more.
Right.
Yeah. I hope you know.
Thank you, Jeremy.
Yeah, of course.
And along that ADC journey, can you share some of your insights into ADC research and development?
Sure. I, glad to have opportunity to share my experience. Mike actually was my ex-boss, and Andrew, we collaborated for many years, and I think in the audience we have, many friends as well. So I'm glad to have opportunity to share the experience with the multinational companies. So when I was at Avipep, we worked with TagWorks and also American company Levena. So using Avipep's diabody and TagWorks the linking payload technology and the Levena as a chemistry. So 3 companies worked together to advance the ADC2 at the moment being approved for clinical trial. So it's great news. And particularly good is about the diabody. It's TAG72 diabody, which is regarded non-internalizing. Okay, at the moment, all ADCs basically rely on fast internalization, non-internalization, people think it's not going to work. So, but this ADC is using non-internalization diabody. So it's called the click release mechanism. So it's different from current or ADCs. So ADC is intravenously injected and then bound to the tumor, and the remaining will be cleared, and then inject a trigger to release the tumor, the drug. Then the drug will diffuse to the neighboring cancer cells to kill cell for the treatment. So, and the collaboration, and led to few, very quality publications. One of actually was voted the best preclinical paper in 2019 ADC World Congress. Yeah, so it's a great outcome for Australian ADC community as well. Yeah, so glad. And also I want the opportunity to promote IDT's Australia, the ADC platform. IDT has been awarded over $1 million fund by the government to establish ADC platform. So we have named the ADC center as the Paul Ehrlich Bioconjugation Center. The center will be equipped with subtle flow expert for gram-scale ADC production. We aim using the IDT's technology to do the GMP production for clinical trials, and also use the facility to collaborate with all institutions, including Mike's company. So, and we are already starting collaborating with multinational companies and institutions. So please, work together. Let's, collaborate. Okay.
Great.
Thank you.
Thank you very much. Thank you very much, Jeremy. We might next to go to Andrew, and I'm interested to hear about what you think the key issues are impacting on bispecific antibodies in the clinic.
Thank you. I guess I can speak in a couple of ways. Firstly, because as was mentioned in the introduction, I actually have a research lab and I've been working with antibodies for well over 20 years now in target discovery and development. And it's been a bit of a journey just with standard antibodies to be able to develop them here in Australia when all of the companies that you're licensed to are actually in US and Europe. In fact, I was reflecting while we were sitting here, I haven't actually got any of the antibodies I've developed licensed in Australia yet, but hopefully in the future that might change. And that's because the biotech industry has really been very focused in the US and in Europe. And as a consequence, you have to very much be aware of what the requirements are by the FDA and the EMEA in terms of the way you develop new molecules, the processes for production, and so on. But I'm also a clinician, and I see oncology patients, I treat them, I run phase 1, phase 2, phase 3 trials. And so I'm quite familiar with what's required to take a molecule into the clinic, what's the minimum requirements as a clinical trialist I would expect to be convinced that I want to take a therapeutic into a clinical trial, because you actually have to convince your oncology colleagues this is worth actually pursuing, because running a clinical trial is a very complex process. It takes a lot of time, takes a lot of resources, and you've got an obligation when you're seeing your patients to try and give them some indication as to whether you really think this might be worthwhile for them. Of course, in early phase studies, you can't have even an understanding of what the therapeutic efficacy will be. But you do want to be able to tell patients, if the data that you've seen is sufficient, that you think this is a study that it would be worthwhile for them to enter. With respect to biospecifics, we've obviously been working on these for many years in the laboratory, but it's only been over the last really 5 to 6 years that we've been studying these in the clinic in any major way. Most of the trials that we've done so far, and that I've been a PI on, which is principal investigator, have actually come from big pharma, and they have typically involved one arm being against an immune checkpoint and the other arm being against another molecule which perhaps may enhance the therapeutic effect of the PD-1 or PD-L1 inhibitor, for example. And what I've found as a trialist and looking at the data that we've been seeing is that very similar issues arise with these newer formats as we were seeing earlier with some of the newer antibody constructs when humanisation techniques were first being developed, which tells you how long I've been doing clinical trials with antibodies. And that is that the preclinical data can give you reasonable estimates as to the safety and the pharmacokinetics in models, but you often see very different data when you go into the patients. And so if you, for example, have pharmacokinetics of an antibody construct, whether it's an ADC or whether it's a radiolabeled version or whether it's a biospecific, you see different pharmacokinetics than what you would expect from the molecular weight. That's where you have sort of the flashing red light in your mind going, there's something happening here. And often what can occur, and this does occur with ADCs, not so much nowadays, but back in the early days, was that you would see rapid clearance. And that wasn't always predicted from the preclinical data. And that was often for ADCs because you had a DAR ratio which induced a positive charge in the antibody. And we have a technique which links into what Mike was talking about, which is where with the very early first-in-human studies, we radiolabel the antibodies to see where they're going. And that gives you information you cannot tell just by looking at PK. And we saw that a lot of these were actually just going to the liver. And that was consistent with the positive charge. And as a consequence, a number of the early generation biospecifics had that issue, that they weren't going where you wanted them to go, or you were trying to tweak two different parts of the immune response, and you ended up having the construct going to the liver or to the spleen rather than to the tumour. And if you know, based on your preclinical models, how much has got to go to the tumour in order to have a therapeutic effect, and you can image it and quantitate it, very early on you can give some guide as to whether or not that's going to work. So I think a lot of the biospecific strategies looking at combined binding to two different immune targets can have unexpected consequences of altered biodistribution, which obviously then means that the impact of the change in the immune system in the microenvironment of the tumour is not necessarily what you want. But I think that we've learnt lessons from some of the early studies, and as a consequence the design of the biospecifics and the way that they're activating is getting better. But every time you come up with a new biospecific design, start again, because it could have the properties that you want or it may not in terms of getting to the target. And then you've got the whole efficacy aspect where a lot of the new biospecifics are trying to reduce the immunosuppressive microenvironment in the tumour while enhancing the PD-1, PD-L1 based effect. And again, whether or not what you can achieve in human can be replicated in appropriate animal models is challenging, and that's where I think some of the new CRISPR knock-in models are very helpful, rather than just the classic, immune deficient or immune competent models that we would typically use in the past. So I think the preclinical data can be very helpful as well.
And since your experience covers these other complex biologics, I'm going to ask you quickly how your drug and radioisotype antibody conjugates can also be developed for clinical use.
Yeah. Well, I think we've had two excellent talks here about how to develop ADCs and also radiolabeled antibodies. From my perspective, they are very complementary, but also very different. Because the way that drugs work can vary from cancer to cancer, and so the type of drug which you give as part of an ADC has to suit the cancer that you're actually targeting, and obviously the target that you're looking for as well. One of the big challenges that we've faced with ADCs of certain types has been what we call the therapeutic window, which means that the difference between the dose at which you start to see therapeutic effect versus the dose where you have toxicity is very small. And it's dose-limiting toxicity which impacts on the dose that you actually give to patients, and that's what your phase 1, phase 2 studies aim to deliver. But I would agree with the comment that the newer forms of drugs which can be used as part of ADCs, particularly the improved linkers, and the stability that can be obtained with the newer production methods is vastly different to what it was 5 years ago, let alone 10 years ago. So I think that if you've got a new target, and really the race at the moment is what are the best new targets, and then you can generate antibodies against them, create ADCs, generate your IP, convince a company that they'll license it, and then charge off into the sunset in your phase 1-2 program and hope that you get reasonable response rates. With radiolabeled antibodies, which is a particular interest of mine for many years, I think that there are huge opportunities, but it's actually taken a number of very, very forward-thinking companies to actually take this forward. And Telix, I think we should all celebrate, because it's really a huge success story for biotech in Australia. I mean, can't keep track of what their market cap is, but it seems to go up every time I have a look. And as I mentioned earlier, historically, biotech in Australia has not been something that's that prominent. You know, you've had the CSLs, but they've been very focused on non-cancer indications. You've had a few others. But there's been, over the last 5 years, an impressive number of companies that have been developing radiotheranostics. I mean, there's at least 5 now in Australia alone working in this space. And also moving more into the other biologic therapeutics, such as ADCs. And so from my perspective, for the very first time, we have an opportunity to engage in both of those areas with Australian companies. That's never happened before. And I think that as these companies grow, they need bright young people to work in the companies that actually know a little bit about antibodies and antibody drug conjugates and radiolabeled antibodies, because they need them. And there's not that much expertise internationally compared to we have in Australia. A lot of overseas companies that work with ADCs or radiolabeled molecules are coming to Australia for their phase 1 and phase 2 studies. And that's because of our favourite regulatory environment, the fact that we know what we're doing, and the Australian dollar. That helps too. So again, there's been an explosion of opportunities to perform clinical trials, and the clinical trial networks around the country are very good. You know, not just Cancer Trials Australia, which is located here in Melbourne as well, that I was chair of for a number of years, but the cooperative groups across a range of different cancer types are very active, and so the ability to create and innovate, patent, license, conduct clinical development within Australia before launching into the overseas markets where you make a lot of the money can still be done. So I think it's an opportune time for everyone who's thinking about where they want to work and the involvement within this field to consider working with antibody or ADC or bispecific technologies.
Yeah, that's why IDT wants to become, first manufacturer for ADC, because we do lots of clinical trial in Australia for overseas companies. If we can do, manufacturing, that would be great for the country.
Absolutely. Thank you, Andrew. Now, Ying, we have next-gen molecules emerging for most of these modalities. I'm interested in your perspective on how platform processes are evolving to support the increasing complexity of bispecific and trispecific and ADC formats.
Thanks for the question. So I think conventionally we use template approach for monoclonal antibody process development. So as we evolve with more complex modalities, I think talking about bispecific, trispecific fusion proteins and ADCs, actually they're very different in terms of process development. So to come up with a new upgraded version of template approach, we always start building upon from our existing knowledge of MAB process, and we will customize certain process steps to fit specific needs for these complex molecules. For example, I think for bispecifics, as I mentioned in my talk, I think using the dual vector approach is almost always very effective to improve the titer expression, as well as solving some problems of misparents. And always we see because bispecifics, they are engineered antibodies, different from nature antibodies. So they always have this tendency to form some aggregates and some fragments problems. So being able to tackle these problems, I think we always will optimize our downstream process, for example, using multimodal chromatography approach to solve some aggregates issue rather than using the traditional 3-step ion exchange approach. I think for fusion proteins, some common challenges we have seen, including, for example, some engineered proteins without Fc fragment. So we will start with coming up with affinity approach for affinity to, if they come up with affinity domains such as other than the protein A, we can tackle the CH1 or CL domain and design those for affinity approach. Or alternatively, if this molecule does not contain any Fc fragment, we can potentially develop template approach using other mode of chromatography such as ion exchange or multimodal chromatography. For example, the emergent VHH minibody, we can potentially use an affinity chromatography to capture a certain group of VHH, or we can come up with some new template approach utilizing ion exchange chromatography for capturing those molecules. And always we see HCP is a big issue for fusion proteins because not only sometimes during the capture step we cannot utilize affinity chromatography, but oftentimes we see some complex interactions between the HCP and fusion proteins, and making those HCP impurities difficult to be removed. So when designing a new process, we almost always should focus on optimizing our downstream process to effectively remove these challenging HCPs. So talking about different type of conjugates, for example, the ADCs, one of the particular concern is how once we have this molecule and specific conjugation, how should we control our CMC process to precisely control the DAR and DAR distribution. And that's almost always a key focus for our CMC development. And also on top of that, how to come up with compatible analytical platform in order to analyze the DAR value, given that's HPLC or mass spec technologies, as well as how to measure the residue levels of different impurities, so that become part of our platform approach. So in general, I would see to tackle those complex molecules, we should always start with lessons learned from MAP and start to build more knowledge from experiences learning with specific challenges with respect to different complex modalities, and step by step upgrade our new template approach so that we can address different type of challenging biologics.
Thank you very much, Jean. And I might switch to Mike now. So you did a good job of explaining these new radioisotopes are emerging and how you're thinking about modifying your antibody to suit perhaps those particular approaches. I'm interested to understand what are the emerging trends for targeted radio pharmaceuticals?
Thanks.
What's next?
Yeah, what's next? So I think for a field that's very much grown up very much around small molecules, and PSMA as being a kind of a darling, with about 50 different types of PSMA targeting molecules as theranostics, including ours. It's about the hunt for new targets, hunt for how do we expand this technology into other diseases, how do we help other patients, because we see that there's still a lot of unmet need there in oncology. We have immuno-oncologies, which is great. And I think ADCs have been a massive impact in some diseases in which they're available and approved. They can have a massive, but then you still get resistance. You still don't necessarily get cures. You still don't get full responses, so complete responses. So there's still unmet need out there, and I think radiopharma has a role to play in those diseases as other diseases. So we're somewhat walking in the footsteps of ADCs. In terms of the targets, but I think there'll be a divergence at some stage with radiopharma, and I think the critical thing for me to understand, or I'd love to know, is where do ADCs and radiopharma drugs, where do they differ and where are they similar? Do we get the same responses? Is it just a different payload? Or do we get, or is it used in combinations, in synergy? Should we use different targets on the same disease? When you have breast cancer, not just HER2 on breast cancer, but maybe there's other targets available, for instance, that you could use as a kind of a two-step approach. Another emerging area, as well as in this very rapidly evolving field, is in the isotopes you mentioned. So alpha emitting radioisotopes. Alpha emitters are very much more powerful, more potent, but they have a shorter range. So the alpha particle doesn't go as far. So it changes the dynamics. Again, it's not just potency, it's also the range at which you use it. And that then dictates which delivery vector do you use. You use a small molecule or an antibody or a mini-body. And I think those are the fascinating areas of emerging trends in radiopharma.
Fantastic. Perhaps we can speak afterwards about thinking about what the radio pharmaceuticals best synergise with and the sequencing of those different approaches. I'm really interested to learn more about that. But do we have time to throw it to the audience for some questions? Or does anyone have a question, a burning question?
Michael, quite a specific question, but it's just really piqued my curiosity. When you get your radio-labeled drug product and you've got like a delivery time of 6 to 10 days to get it to the hospital or the patient, how does that work with, say, release testing? And obviously that's highly expedited release testing and things like sterility testing, which usually have quite long lead times.
Yeah, great question. I was going to talk about that originally and I kind of steered away from it, but you're actually doing the release testing. You have you work on the process validation becomes really important because a lot of your release tests are done like sterility is done retrospectively after the patient's been dosed. So again, you're validating the process of keeping something sterile, So, and we typically have terminal filtration on each dose anyway. So, all of these are having process steps and validation of process and training and all that sort of stuff that goes into GMP. So, it's one of the oddities about sterility testing because even if you had a really rapid method to test for sterility, you'd have to have somewhere that has a radiation license to handle it. So not many pathology or not many testing centers want to handle. We have to, during the process validation, we have to let them decay and then send them out once they've decayed to a certain level and then see if they're everything. So it's one of these little oddities that are quite difficult as well.
I might just comment that sterility testing in the way that Mike described as being retrospective is what we do for all diagnostic radiopharmaceuticals. I mean, in my department we have our own cyclotrons, so we make all our own FDG gallium-based peptides. Around Australia, I think last year there were almost 250,000 FDG PET scans performed. Not a single one of them has sterility done before they're injected, and you'd have possibly 1 or 2 reported adverse events per year. So again, it's all based on the process itself, and that's completely accepted, particularly with short half-life isotopes, that you don't have to do it before release, and typically you have to wait until everything is decayed before you actually do the sterility testing anyway, so that's always a bit of a challenge.
I'm going to skip over my usual plugs, except to say keep an eye on our website and our emails for all of the upcoming events. We would love to see you there. If you haven't already, please do join Willem as well, which is our online community, which is great for connecting when we can't do it in person. So everybody online, it's really good to connect them with them as well. And you can share your successes and the opportunities and engage digitally too. So thank you all so very, very much, and for your patience as well. Tech is great when it works perfectly, and those small little increments can feel like a lifetime, can't they? So thank you all so much. Thank you to RSM, and there is canapes and networking just outside. Thank you.